So in this first session, these are the topics I will cover. An introduction to cancer as a disease caused by damage to the cell's DNA and the types of damage we see in cancer cells, but also I will touch on the dual reasons why our risk of cancer increases as we get older, that it's not just about mutations, but it's also very much about our immune system and how our immune system normally patrols the body and eliminates cancer cells. I will then turn to the way cancers evolve and diversify over time and why that is such an obstacle to curing cancer patients, along with the way that cancer can spread around the body. But I'm going to begin where I think we often begin, with cancer as a disease caused by damage to our cells' DNA. So we'll begin with our cells. So our body contains trillions and trillions of these tiny little structures, the living cells of our body. And within those living cells are structures called chromosomes. And within those chromosomes, that's our cell's DNA. So the DNA is a long spiraling molecule packaged up and condensed down to form these structures called the chromosomes. And then those chromosomes are inside the nucleus of each cell. So virtually every cell in our body has the same set of chromosomes and contains the same amount and same sequence of DNA. And within that DNA, there are lengths of DNA that the cell can recognize as being different from the rest. And it can tell that this bit of DNA, unlike most of the cell's DNA, is a gene. It is a set of instructions telling the cell how to make a particular protein. And the proteins our cells make, there's tens of thousands of different ones. They come in all sorts of different shapes and sizes. They are constructed from tiny little chemicals called the amino acids, and the order of those amino acids is what is determined by the information in that gene. Within our cells' chromosomes, there are about 20,000 genes containing the instructions for making all the different proteins our cells need. And it's the combination of proteins that the cell makes that then dictates that cell's behavior. The genes, the DNA, are the library of information for the cell that the cell keeps looking back at, keeps checking back, keeps reading and using as a source of information, as a library of information, telling the cell how to make lots and lots of different individual proteins. And it's the proteins that do all the work of the cell. They're the things that make proteins. They're the things that transport things around, that connect the cell to its neighbors, that give the cell its shape and its structure, allow it to perform chemical reactions. All the work of the cell is done by the proteins, and the proteins are manufactured according to the instructions found within the cell's genes. If a gene is damaged, another word for that being mutated, then instead of making the normal healthy version of the protein from that gene, the cell might make a faulty version of the protein from that gene. And I'm imagining here that the damaged gene is the gene for making a cell surface receptor protein, and that when that cell surface receptor protein is damaged, that causes the cell surface receptor to continually send signals into the cell, telling it to grow and survive and multiply, and that cell is now part way to becoming a cancer cell. So our cells all contain the same DNA. They're using that DNA to make all the proteins they need. The exact repertoire of proteins that they make will be different in each type of cell. You know, a lung cell is going to be making different proteins to a skin cell or a nerve cell or a kidney cell. A lot of the proteins will be the same because they're responsible for just the general functioning of every cell, but some of them are specific to individual cell types. But the repertoire of proteins a cell is making, how much of each one, where they're located, what they're doing, that dictates the cell's behavior. And so if a gene becomes faulty, if it's damaged, if it's mutated, and the cell makes a faulty version of a protein, it's going to start behaving abnormally, and it may start taking on some of the properties of a cancer cell. There's very few instances where just one mutation can cause a healthy cell to become a cancer cell. Usually, cancer is caused by multiple mutations in multiple genes. Perhaps the first mutation was one that affected one of these cell surface receptors, and it forced the cell to grow and multiply a bit quicker than its neighbors, a bit more likely to survive than its neighbors, and so it's multiplying a bit faster than normal and it's a bit unstable. Then the cell picks up a second mutation, and this means it's unable to die. Some of its normal checks and balances have now gone, and even though it accumulates even more damage over time, that cell can't die, and also it goes undetected by the immune system because there's a mutation affecting the relationship between the cell and the immune system. This is a hypothetical sort of scenario. Usually there's a few really important driver mutations forcefully causing the cells to behave as cancer cells, and then a whole load of lesser mutations adding to that mix and adding to those behaviors. Scientists over the years have tried to pinpoint what are the behaviors of cancer cells that set them apart and make them different from the healthy cells of our body. And they've come up with this list of characteristics that cancer cells exhibit called the hallmarks of cancer. Now, this list is fluid. Scientists keep adding to it and expanding what they mean by some of these hallmarks and tweaking it. And every 5 or 10 years or so, there's a new version of the hallmarks. So this is a simplification. I'm not trying to encompass all the work that's been done to characterize these hallmarks, but I think it's a useful idea, a useful framework to give us some insight into what makes cancer cells different. So they tend to contain mutations affecting proteins that then force them to multiply in a way that normal cells don't. Normal healthy cells only multiply when they are given the instruction to do so by neighboring cells, whereas cancer cells can give themselves those self-same signals. So when there's an overactive receptor protein, for example, on the cell surface, that's generating signals for the cell, which in normal circumstances would come from neighboring cells, but now the cell is generating those signals for itself. They tend to be more resilient and less sensitive than healthy cells, that a level of DNA damage found in cancer cells would cause healthy cells to die. So those normal processes that cause faulty cells to die have been affected by mutations. They avoid destruction by the immune system. This is something I will keep coming back to in the coming weeks because it underpins our the creation of various immunotherapies, that cancer cells often go undetected by the immune system because of some of the mutations they contain. They have an unusual ability to multiply. Healthy cells will only multiply a certain number of times before they become dormant and they will never multiply again. There's an internal clock that dictates how long our cells keep multiplying for and when they stop. Cancer cells reset that clock and they gain an endless capacity to multiply that healthy cells don't have. They create their own blood supply, either by producing molecules which trigger the growth of blood vessels or by co-opting whatever blood vessels exist in that tissue and growing along the length of the blood vessels. So they can do this in a few different ways, but they will always need some sort of blood supply. They become insensitive to negative signals from their neighbors. Their neighbors, even inside tumors, are trying to send restraining signals to cancer cells, but they gain the ability to ignore them. You tend to find inside tumors lots of white blood cells that you would normally think of as being involved in inflammation. Things like macrophages and neutrophils and eosinophils, various different white blood cells that when present for short periods of time in response to an infection or damage, is a healthy thing. But if permanently located inside a cancer, inside a tumor, this becomes a bad thing. They actually become part of the problem. They send out nurturing and supportive signals to cancer cells. They have altered energy metabolism. They tend to switch to an inefficient way of producing energy. They're very good at grabbing the energy they need, and they use it in this inefficient manner because that also produces complex molecules that they need for further growth and proliferation. In order for cancer to become a life-threatening disease, often that change from it being a just a single cluster of cancer cells or a single tumor, what makes it life-threatening is when cancer cells break away, squeeze into blood vessels or lymph vessels, and travel elsewhere in the body and create metastases. This is when it becomes a life-threatening disease. And then lastly, they are genomically unstable. This is the fancy way of saying they are prone to accumulating more and more mutations over time. They tend to be quite bad at detecting DNA damage and very bad at repairing DNA damage. So they accumulate a lot of mutations at a much faster rate than a healthy cell would. So these are some of the properties of cancer cells that are caused by the DNA damage that they contain. And that DNA damage, a lot of it is unavoidable. It is damage that occurred during the early stages of embryogenesis, where lots and lots of cells were multiplying very quickly. You can inherit damage. You can inherit gene mutations from your parents and grandparents. They can be passed down through families. But a lot of it accumulates as we go through life. Our cells are performing thousands of chemical reactions. They're really busy. They're doing lots of stuff. And a lot of the chemical reactions they perform produce byproducts that cause DNA mutations. So there's always going to be, every day, every cell in our body, there's going to be mutations accumulating in their DNA if they don't manage to repair all the damage that's taking place. And they never manage to repair all of it. So there's damage that gradually accumulates, there's damage that we inherit. Also, as cells multiply, they always make a few mistakes in their DNA when they go through that multiplication process. So there's a whole lot of mutations happening every cell, every day in our body that we can't influence, that we can't change, that are a fact of life. But then, of course, added to that are the things we're exposed to, the lifestyle choices we make, our diet, what exposures we are come across in terms of infections and disease, the cycles of hormone production. All of these things also influence our cancer risk. Now, they may be potentially avoidable. I think for a lot of the time they are not avoidable. You know, if you are somebody growing up in a poor area of India, you are going to be cooking your food over a fire in your house. There is going to be smoke. There is going to be a lot of air pollution that you cannot avoid. So for each one of these exposures and even choices, there may be limited things we can do to avoid those potentially avoidable causes of DNA damage. It all depends on who you are, where you are, what options are available to you, what influences are being exerted on you. The types of DNA damage we find inside cancer cells are very varied. You can have massive chunks of chromosomes broken, rearranged, accidentally added to. All sorts of things can happen to a chromosomes on a macro level. There are also slightly smaller scale mutations where just chunks of chromosomes have been deleted or accidentally recopied several times, inverted. A lot of this kind of damage happens to cells as they are multiplying. When a cell multiplies, it has to make a duplicate set of all of its chromosomes. It lines up all those duplicated chromosomes on a structure known as a spindle, and then the spindle fibers retract and the chromosomes are pulled apart so that one copy of each chromosome ends up at either side of the cell. So when the cell cleaves in two, you've got a full set of chromosomes for each of the new cells produced. A lot of problems can occur during that process of the fibers contracting and the duplicated chromosomes being pulled apart. This is where things can really go wrong, of one cell ending up with duplicated chromosomes, one cell ending up with missing chromosomes, things get broken up, they get swapped around. That's often the point in time where the large-scale chromosome damage occurs. The much more subtle damage, just involving a few tens or hundreds of DNA bases, the indels, the small-scale insertions and deletions, and the point mutations that just affect a single DNA base, a single chemical component of the DNA molecule. These are less associated with cell division. They can happen through all these different chemical reactions and different processes our cells are undertaking. And then there are also epigenetic alterations that we find in cancer cells. So normally in cells, their DNA is packaged up very tightly, and to facilitate that maneuvering of the DNA and facilitate its packaging up or being relaxed, that is done by adding bits and removing things to the backbone of the DNA molecule. Methylation, one carbon and three hydrogens attached to the DNA, that's called methylation. That tends to make the DNA more compacted. A slightly different chemical modification called acetylation tends to make the DNA molecule more relaxed. The more relaxed and open the DNA is, the more accessible the genes are, the more protein is made from those genes, the more compacted the DNA is, the more inaccessible the DNA is, and the less likely it is that that bit of DNA is being used to make proteins. So epigenetic modifications or epigenetic alterations, we find these in cancer cells. The wrong bits of the chromosomes are relaxed, the wrong bits are tightly coiled. The cell is therefore making the wrong amounts of different proteins, and that's influencing the cell's behavior. I'll just give you a few examples. So chromosome translocations are when two chromosomes break. The cell tries to repair things and put them back together, but it gets things the wrong way around. You might end up with two genes from two different chromosomes ending up on one chromosome, and instead of making two separate proteins, the cell makes one long fusion protein. An example being the BCR-ABL fusion protein we find inside the cells of chronic myeloid leukemia. Another type of damage is when you get extra copies of a gene made. We're supposed to have two copies of every gene because we inherit one from our mother and one from our father. But sometimes in cancer cells, you find there's extra copies of a particular gene or group of genes. An example being the ERBB2 gene for making the HER2 protein in breast cancer. In about 20% or so of breast cancers, there's extra copies of that ERBB2 gene, meaning the cell is making extra amounts of the protein from that gene, the HER2 protein. The HER2 protein is one of these cell surface receptor proteins, and it gives a strong signal to the cell to grow and multiply and survive. A much more subtle type of mutation is when you get just a single DNA base, a single DNA chemical swapped for a different one. There are 4 DNA bases. These are tiny little chemicals that span the gap between the two strands of the DNA double helix, and they always pair up in a particular way. A pairs up with T, C pairs up with G. And just occasionally, even a single base change, in this case from a T to an A, causing a single amino acid to be altered in the protein made from that gene. And in this case, rather than the normal valine amino acid, you get a glutamic acid amino acid being put in place. And this forces the normal BRAF protein to become a hyperactive, cancer-causing version of the BRAF protein. So in cancer cells, you tend to find a mixture of DNA damage. You find large-scale damage, you find smaller-scale damage, you find the smallest scale damage of individual base pairs that have changed, you find alterations to the surface of the DNA molecule, and you tend to find a mixture of lots of different types of damage in cancer cells. And scientists over the past 10 years or so have been trying to identify patterns of damage that they can attribute to particular causes. So they've discovered patterns of damage that only seem to occur if you have a history of smoking. Patterns of damage that only occur if the cancer cells contain a mutation in a BRCA gene. Patterns of damage associated with overactive ApoBec enzymes. These are natural enzymes in our cells, but cancer cells often contain overactive versions of them, and that creates its own pattern of damage. So they've now discovered well over 100 different patterns of damage that can be linked back to specific causes, or in some cases, we don't know the cause. We know that there's a particular pattern of damage, but we don't know where or why it happens. And when somebody develops cancer, their cancer cells will contain multiple different patterns of damage that can be attributable to different causes. Some of those patterns will probably be linked to age. They might be linked to a problem with DNA repair. They might be linked to reactive oxygen species, which are produced by a number of different chemical reactions. They might be caused by overactive ApoBec enzymes. So for each person, if you really wanted to, you could be like an archaeologist looking at the DNA damage in their cancer cells and working out the natural history, the life course of that cancer. In what order did the mutations happen? What caused the mutations to occur? What patterns of damage do you find in the person's cancer cells? You could do that kind of analysis for each cancer patient, but you'd spend the whole NHS budget doing that. So we don't do that because it would be hugely costly and time consuming, and it wouldn't necessarily tell us how best to treat that patient. So we reserve our tests and our analysis for looking for specific types of damage or specific genes that have been damaged, where we can match that up with a treatment that's likely to be effective for the patient. You know, if we find extra copies of the ERB2 gene and extra amounts of the HER2 protein, we know that person is likely to benefit from a HER2 targeted treatment. If we find that point mutation affecting the BRAF gene, we have BRAF inhibitors. We can offer that patient a BRAF inhibitor. We don't do massively in-depth analysis unless there is a practical outcome of that, unless it changes what we're going to offer that patient. So we can work out the life history of that cancer, and scientists do this kind of analysis, but it's a research tool, and it's very interesting to know what sorts of patterns and what sort of damage you find in cancers, but it doesn't necessarily help the patient to have done that kind of analysis. Now, I'm sure it's of no surprise to anybody that our risk of developing cancer increases as we get older. And some of that increase is because as we get older, our cells have multiplied more, they've made more mistakes, they've accumulated more damage, we've been exposed to more things. The amount of DNA damage in our cells does accumulate over time as we get older. So as we age, it becomes more and more likely that a cell will have accumulated damage that is now forcing that cell to behave as a cancer cell. However, most of the time, that doesn't lead to a diagnosis of cancer, because most of the time, our immune system steps in and destroys the faulty cells. But as we get older, our immune system often becomes less efficient. The immune cells become less vigilant, less able to detect cancer cells, and even if they should find them, less able to destroy cancer cells. So neither of these things on their own is sufficient to cause cancer. We're not going to get cancer just because our immune system becomes less efficient. And we're not going to get cancer just because a cell accumulates mutations, because that cell should be destroyed by our immune system. But as we age, it tends to happen that cells with mutations accumulate and our immune system is less likely to destroy them. So whereas at the age of, in our childhood and early 20s, the cells of our body are relatively free of mutations. But as we get older, patches of mutated cells start to appear. And by the time we reach 70 years old and beyond, our whole body is a patchwork of populations of faulty cells. I think, whenever I look, well, increasingly at the backs of my own hands, but very differently when I look at my mum's hands, she's 83 now, I think. The back of her hands is just a mosaic of patches of skin with slightly different pigmentation to them. That is an outward sign of a process that's going on throughout our whole body, internally as well as externally. Our whole body is a patchwork of cells with slightly different properties, some of them growing slightly faster than others, some slightly more resilient than others. There is not an inevitability to cancer, but it is so linked to that aging process that as time goes by, more and more populations of faulty cells appear, and our immune system just becomes less able to destroy those faulty cells and less able to destroy the cancer cells that occur. There are things we can do to help our immune system remain as vigilant as possible. Exercise, good diet, these things really do have an impact on our T cells, which are the ones most likely to be on the lookout and destroying cancer cells. Also, the more, if you're exercising regularly, if you're keeping your body moving and active, your organs and tissues are less prone to inflammation. You can help keep your body hostile towards cancer cells by keeping moving and keeping physically active. So just to underline that relationship, that because DNA mutations accumulate as we get older, because our organs and tissues and immune system change as we get older, our risk of cancer does gradually increase. One of the most dangerous and difficult aspects of cancer is the way that the individual cancer cells pick up more and more mutations over time. And that means increasingly the cancer cells become diverse. So that initial cancer cell, the first one that had accumulated mutations to a point where it's now behaving as a cancer cell, that probably only had a handful of cancer-causing mutations. But that cell multiplying faster than normal, not having as many checks and balances as normal, it was unstable and it was prone to accumulating more mutations over time as it multiplied. And so each new cell has that property. It tends to be bad at detecting DNA damage, bad at repairing DNA damage, and likely to pick up more and more DNA damage as time goes by. And so the clusters of cells within tumors gradually become more diverse as time goes by. In part, it will depend on how long this process has been going on. So, for example, bowel cancers, often there's a long history of the accumulation of mutations over several decades. So there's been a long time for this diversification to take place. Equally, multiple myeloma, a cancer caused by antibody producing plasma cells in the bone marrow. From what we can tell, often that multiple myeloma, which might be diagnosed when you're in your 70s or 80s, those multiplying antibody producing cells are producing antibodies to an infection you were exposed to maybe 30 or 40 or 50 years ago. That antibody producing cell in the bone marrow has sat there multiplying very slowly, producing just a bit of antibody, and it's just kept going and going and going and slowly accumulating more mutations, multiplying. And so the disease, multiple myeloma, is so hard to cure because there's so many different pockets of cells with their own unique combinations of mutations, their own behaviors, their own sensitivities to treatment. But we see this time and again, particularly in advanced cancer, that whatever treatment you give, it hasn't been able to eradicate every single cancer cell. It's eradicated some cells, the tumor has shrunk, the tumor might have even disappeared off the scan. But because there's so much diversity, there's a few cells that have managed to survive treatment, maybe because of the mutations they contain, maybe because the environment they've created from themselves, maybe because of support and protection they're receiving from the cells in their vicinity. Cells that survive and continue to multiply are the cells that cause recurrence. And cells that cause metastasis, often they are breakaway cells from a primary tumor that maybe in the primary tumor are now completely undetectable. They've been overgrown by other cell types. They may have completely died off in that primary tumor, but having broken away from the primary tumor and reached new locations and started to multiply, they create a metastasis. So you can't presume that the cells in a metastasis are going to behave the same and have the same sensitivity to treatment as cells in the primary tumor. Apologies, my dog is going a little bit mad because my husband's just arrived home from work. So there's diversity amongst the cells in the primary tumor. There's possibly various different metastases, all containing different populations of cancer cells to varying proportions. And it means that whatever treatment we give, there are going to be cells that manage to survive. And this is going to be true particularly for something like a targeted therapy, where we have identified maybe a BRAF mutation, a HER2 amplification. We've identified a particular mutation that we can match with a treatment, but we're targeting one mutation with one treatment. And because of diversity amongst the person's cancer cells, if they have advanced cancer and we're targeting one mutation found in one biopsy from one part of a tumor, it becomes increasingly unlikely that we will be able to cure the patient with that one treatment. A treatment like chemotherapy, which is toxic to pretty much everything that's trying to multiply or trying to stay alive, that toxicity of chemotherapy is a bit more general. It's a blunt tool. You're affecting lots of different populations of cells. So chemotherapy can be a useful option, especially when it's a very diverse and varied population of cancer cells in the person's body. A targeted therapy might have greater initial impact because you're killing a lot of cells very quickly, but over time, if they have advanced cancer, it's highly likely that cancer will re-emerge. Immunotherapy is different again because the immune system can cope with diversity. It can single out cells and destroy cells with different properties and different mutations. So immunotherapy does have the possibility of curing somebody even with advanced disease, even with varied populations of cancer cells. Immunotherapy can still only cope with a certain amount of diversity and variation. There does come a point where the immune system just gets too confused and bamboozled by the amount of diversity exists, but it can be effective against tumors that are more diverse, that you'd see rapid resistance with a targeted therapy. So this is one of the most difficult parts of curing anybody with cancer, particularly advanced cancer, is that the more diversity that exists amongst the cancer cells, the less likely it is that you're going to be able to cure that person of their disease. And as I'm sure you've experienced and something that you know full well, the more different treatments the person has had, the less of an impact those treatments seem to be having. That the first treatment the person receives is generally the most effective, and then the second course of treatment they receive at the point of relapse tends to be less effective. That might work for a little while, and then the cancer accelerates again. The next treatment you try and give, that's even less effective than the last time. What tends to be happening is with each course of treatment, you are killing off the weaker cancer cells, the less protected cancer cells, the more vulnerable cancer cells, and you are leaving behind the hardier, more resilient cancer cells. So you might find that at the point of diagnosis, you don't detect cells containing a TP53 mutation. But TP53 mutations tend to make cancer cells more resilient, less able to die, less sensitive to treatment. And so if you give the patient treatment and then their cancer reemerges, that is when you tend to find that the cancer cells with TP53 mutations are now dominating, because these are the ones that were able to survive treatment, whereas the weaker and more sensitive ones couldn't. So the more time goes by and the more treatments the person has received, the more you find cancer cells with these sorts of mutations that make them very resilient, that make them very unable to die. And of course, another important problem facing us in somebody with advanced disease is that their cancer has spread, that you're no longer treating a cancer that's just a single tumor, a single population or a single unit of cancer cells, but there are now cancer cells in multiple locations in the body. And what seems to happen is in the primary tumor, not all cancer cells are equal. There's diversity amongst the mutations they contain. There's diversity in terms of their access to a blood supply or their access to lymph vessels. There's diversity in terms of the types of white blood cell found in different parts of a tumor and what they're doing there and how they are interacting with the person's cancer cells. And so you end up with each individual cancer cell being unique in terms of its mutations, in terms of its access to oxygen, access to nutrients, access to blood supply. Each cell is unique. And some cells are more resilient, more attached to one another, less attached to one another. And cancer cells can change, not just because of accumulating more mutations, but also as they respond to changes in their environment. If oxygen levels drop because the blood supply is compromised, that will change their behaviors. If the pH drops and the environment becomes more acidic, that will also change the cell's behaviors. So a combination of the mutations the cells contain, the white blood cells in the environment, change in the cell's environment in terms of oxygen and pH, can lead a cell to become more resilient, less attached, more mobile than its neighbors. And that cell detaches from its neighbors, the normal proteins that connect cell to cell, those dissolve, they're destroyed. The cell releases enzymes that digest proteins that stand in the way of the cell accessing a nearby blood vessel. And it also releases molecules that trigger the blood vessel to become more permeable. The cancer cell is able to squeeze inside the blood vessel. Now, millions of cancer cells might make it into a blood vessel, but only a tiny fraction of the ones that make it into a blood vessel are going to survive. The blood is a very turbulent place. There's lots of red blood cells battering into the cancer cells and destroying them. They get squeezed. They are not adapted to life inside a blood vessel. And so most of them will die. But if just the occasional cancer cell manages to travel to a new location, survive, stick to the inside of a blood vessel, reverse the process of squeezing through the blood vessel and ending up into the tissue, that cell may cause a metastasis. It still probably won't. Our body is designed to be hostile towards cancer cells. But it does seem like the cells in a primary tumor can emit molecules that affect distant organs and tissues and can actually prepare the way and make those distant organs and tissues less hostile to any cancer cells that end up there, and ultimately allow those traveling cancer cells, those cancer cells that have moved to a new environment, allow them to multiply, attract in white blood cells that facilitate their behaviors, white blood cells that nurture and support them and protect them and enable them to create a new secondary tumor. Whether or not a cancer spreads has an enormous impact on whether that person can be cured of their disease. And whether or not their cancer spreads is going to be determined by a combination of different factors. What is the individual genetic makeup of each cancer cell? Are there cancer cells that have the innate resilience and ability to multiply and ability to populate a new organ or tissue? Do those cells exist? Are they near enough to a blood vessel or a lymph vessel or even a nerve that they can infiltrate and then travel to a new location? When they get to that new location, are they able to alter that new environment in such a way that that cell is now able to multiply and create a new tumor? Of course, there's always interest in creating treatments that prevent metastasis, but the reality is that for most people, by the time they are diagnosed, the die is already cast as to whether their cancer is likely to spread or not. We know for the vast majority of people who have operable pancreatic cancer and receive surgery, even though you might have clear margins for the surgery, the vast majority of those patients are going to relapse. We know with breast cancer, with hormone-sensitive breast cancer, that even if it seems like it's been cured and the person seems to be free of disease, it can still return 10, 20, 30 years later because cancer cells broke away long before the diagnosis and they've been sitting dormant in the brain or the bone marrow or other protected places, and then at some stage they reactivate. So although the idea of preventing metastasis with treatments seems very appealing, we've never managed to do it because probably by the time we're treating the patient, whether their cancer is going to spread or not is already determined by what has happened prior to that diagnosis. So I'm going to summarize what I've covered in this session. I started off by saying that the DNA in our cells is a bit like an inner instruction manual for the cell. It's a library of information that the cell returns back to over and over again, because within that DNA are the instructions for making every protein the cell will ever need to make. And different cells, different types, they look different, they behave differently because each one is accessing a slightly different combination of genes and making a different combination of proteins. The DNA in our cells is constantly getting damaged. This is a daily occurrence. Scientists believe that in each cell, every day of our lives, its DNA is getting damaged 70,000 times. So for each of the trillions of cells in our body, each one is sustaining damage to its DNA 70,000 times a day. That's a huge amount of damage. Our cells have all sorts of DNA repair proteins to repair that damage, but there's always some that's going to get missed. So there is an inevitability to the accumulation of damage in our cells. But it doesn't mean that cancer is an inevitability because our immune system is there to protect us. It's constantly vigilant. It's constantly on the lookout. Cells like our T cells, our macrophages, our natural killer cells, they're constantly watching and destroying cancer cells that emerge. So cancer occurs when two things have happened. When a cell has accumulated damage to the point it's become a cancer cell, and for some reason that cancer cell has not been detected and destroyed by the immune system. And this tends to happen as we get older. Mutations accumulate, and our immune system becomes less vigilant. Something I'm going to come back to next week and next time and in the coming sessions is the way cancer cells interact with our immune system, and they are very much affected by our immune system and they very much influence our immune system. And this is the big focus of immunotherapy, of how to take advantage of that increasing knowledge of the interaction between cancer cells and white blood cells. And that is leading to a lot of new treatments. What though is still a massive obstacle to us, and an obstacle we are as yet unable to overcome, is the way cancer cells evolve and change over time. This problem of intratumoral heterogeneity, the diversity amongst the cancer cells in a person's tumor, not just in terms of the mutations they contain, but also in terms of variations in their physical environment and physical makeup. This degree of variation that we find inside tumors is an obstacle we've not yet been able to overcome. We don't have the treatments that can really tackle that. Nor do we have treatments that can prevent metastasis. This, again, currently enormous obstacle to successful treatment is the way that cancer cells not only evolve and change over time, but also spread to new locations in the body. So next session is all about targeted therapies, and these are the treatments that have been developed through an understanding of the mutations that cancer cells contain and the influence those mutations have over the cell's behavior. So it's things like the cell surface proteins and the enzymes inside them. As we identify mutations, as we identify differences between cancer cells and healthy cells, we can develop treatments that take advantage of that knowledge and exploit those differences. The immunotherapies that I will then come to in subsequent sessions, these are the treatments that take advantage or build on our increasing understanding of how cancer cells interact with the immune system and how we can develop treatments to help the immune system attack and destroy cancer cells. I am going to stop there and I know we're a small group, but I don't know if there's anybody who wants to ask any questions about the content I've covered so far.
No questions, but as always, makes it really easy to follow. And I like your graphics. Your graphics actually make it really easy to follow. So you're really good.
That's good. I spent a long time, me and PowerPoint, creating new pictures.
Yeah, I agree. I've not attended one of these before. very easy to follow. Thank you.
Good, good. Well, I mean, hopefully this first session is the kind of the preamble and the fundamentals that then mean the subsequent sessions people get more from them. Because I think unless you understand the mutations, you're never going to understand the targeted therapies. But I think also you're never going to understand why a targeted therapy can't cure somebody with advanced cancer, or it's very unlikely to, just because of this problem of diversity that really, we're making baby steps, but I've yet to see anything where you go, oh, okay, great, we're going to make inroads into this. We're going to have treatments that can actually deal with that level of diversity. Yeah, it still just seems a huge mountain of an obstacle in our way.
I think as well, the way you describe it makes it easier for CNSs and people who are not day to day in science to explain it to their patients. So when patients kind of say, well, but why are you not telling me it's going to cure it? It's nice for us to have the language to explain it to them, which is good.
Yeah, I think it makes sense of a lot of things, I think. The issue of diversity.
Yeah. So yeah, no other questions from me. Thank you.
Is our next session, is that in a couple of weeks?
Something like that. I'm not entirely sure what the date is. I think it's something like 2 weeks.
Just quickly check.
Spread them out every 2 weeks.
Yeah. Okay, so it's
Monday the 9th.
Monday the 9th of March. So that's when I'll look at the targeted therapies.
Okay.
So yeah, if you wouldn't mind sending us out the titles again, and then we
Yeah.
keep them changed.
Yep.
Yeah, we'll do.
I'll
Because I think
resend you
some
the.
people will dip into particular ones more than others, and then our
Well, they
new
might have
staff are going to
missed
get them to
certain
sit all of them.
sessions the first time round.
Yeah,
Yeah.
exactly.
Okay. Yeah, I will do. I'll resend you the program just so make sure you have that.
Lovely. Thank you. Well, have a good evening.
Thanks very much.
Okay, and
Yeah.
you.
All right. Take care.
Take
Bye.
care. Thanks.
Bye.
Bye. Bye.